Hello friends, so today we are going to discuss topic Kelvin cycle. So this topic belongs to photosynthesis in higher plant. Actually this topic explain about the photosynthesis mechanisms. So actually two type of reactions occur in the photosynthesis in higher plant. One is the light reaction and another is the dark reaction. So light reactions, there is a production of ATP and ADP, NADPH. In dark reaction, there is a utilization of ATP and NADPH. So by utilization of ATP and NADPH, so but there is a production of the glucose. So Kelvin cycles, this cycles has been explained by Melvin Kelvin. Kelvins. They explain by the help of C14 radioactive technique. Radioactive technique. So Kelvin explain how light reaction light reaction convert into chemical reaction. C3 cycles, it is Kelvin cycle also known as the C3 cycle. Why we call Kelvin cycle C3 cycle? Because after the carbon fixation there is a first stable fixation, there is a three carbon containing compound, that is a phosphoglyceric acid, 3 PGA product, and phosphoglyceric acid that contain three carbon compound. So that's why we call this three C3 cycles. C3 cycles, that is also known as the dark reaction. Dark reaction. Why we call C3 cycles is dark reaction? Because C3 cycle is a light independent reaction. It is light independent reaction. Light independent reaction. So why C3 cycles known as independent reaction? Because during the process of these mechanisms, there is no requirement of light. But actually, this cycle is depend upon the product of light reaction. So this cycle depend upon the product of light reaction. This cycle depend upon product of light reaction. Light reaction. So now we are going to explain about these cycles in detail. So first of all, what is requirement for this cycle? So one is the Rovisco. Rovisco, this is the most abundant enzyme in this earth. Abundant enzyme in this earth. So Rovisco full form ribulose bisphosphate, carboxylase, and oxygenase. This is the full form of Rovisco. So how they work, we will see their function further. Substrate, two type of, there is a substrate, that is a RUBP substrate used during the C3 cycle. Ribulose, bisphosphate, 1,5 bisphosphate, 1,5 bisphosphate, there is a substrate. So basically, what is the aim of this reaction? Kelvin cycle. So aim is production of glucose. How this cycle occur? So actually, what happen when, first of all, we explain the nature of Robisco. When in the medium, when in the medium, O2 concentration is greater than CO2, then RUBP, Robisco, prepare oxygen. Second conditions, when in the medium CO2 concentration is greater than O2, then what will happen? Then Robisco prepare CO2 then oxygen. So these things has been explained why these things has been explained by Warber, scientist Warber. So that's why this is known as Warber effect. Warber effect means they are giving preference to the on the basis of the concentration of gases, Rubisco. So if we tell that where this reaction occur, so C3 cycle occur inside chloroplast, of mesophil cell, chloroplast. Basically, we can say it is occur inside mesophil cell. We can say it is occur inside mesophil cell. So Now we are going to discuss the stage of the C3 cycle. So there is a three step stage in the C3 cycle. Stage of C3 cycle. So basically there is a three type of a stage present in the C3 cycle. So first stage is carboxylation. That is carbon fixation. Second is reduction. Reduction of ADP, ATP and NADPH. And third stage is regeneration. By utilization of ATP. Utilization of ATP again RUBP regenerate. So now we are going to discuss about the C3 cycles. So this is the basic plot of the C3 cycles. So now we are going to discuss. So in the medium, in the medium, carbon dioxide is greater than, concentration of CO2 is greater than oxygen, then RUBP prefer CO2. Rubisco. So Rubisco enzymes brings, and there is a first stable product, that is a used as a substrate. This is a RUBP, that is a 5 carbon containing compound. RUBP. Now utilization of the CO2, and utilization of the CO2, this is helped by the Rubisco. So there is a how much CO2 they utilize. So if we tell about the number of CO2, approximately 3 CO2. So now there is a first stable product, that is formed during this reaction, that is a 3 PGA, phosphoglyceric acid. So 3 PGA phosphoglyceric acid, sorry. That is a 2 PGA, and it contains 3 carbon, means total 6 carbon. So now there is a reduction occur. Reduction. So this is a carboxylation process, is carbon fixation, carboxylation, and due to the carbon oxidation there is a first stable product, that is a 3 carbon containing product, and after there is a reduction, and reduction of 2 ATP, that reduce ADP plus IP, inorganic phosphate. And now after the reduction, 1,3 bisphosphate, bisphosphoglycerate, phosphoglycerate. So now 1,3 bisphosphoglycerate, they will convert into the 2,1,3 because bisphosphoglycerate also contains 3 carbon containing compound. Now again there is a also reduction reduction of the 2 NADPH. So once it's reduced, get reduced 2 NADP plus H plus 2H plus. So now 2H plus is forms. So 1,3-bis phosphoglycerate, that is convert into the glyceraldehyde 3-phosphate. Glyceraldehyde 3-phosphate convert into the glyceraldehyde 3-phosphate. And now glyceraldehyde 3-phosphate, that is a 2 carbon, there is a 3 carbon containing, there is a 2 glyceraldehyde 3-phosphate is forms. Now they utilize 1 carbon atom, 1/6 carbon C6H12O6, and remaining 5 carbon they undergo for regeneration. There is 1 carbon utilized for the glucose production, and remaining 5 carbon go for glucose ATP RUBP regeneration. So this is the ADP plus IP. So this is the regeneration. This is a reduction phase. This is a reduction phase. So now we can see there is a reverse glycolysis form, and after reverse glycolysis 1/6 part of glucose formation occur. So reverse glycolysis, how it will form? 3 glyceraldehyde 3-phosphate, that is converted to the fructose 1,6-phosphate, then it is converted to the glucose 6-phosphate, then fructose 6-phosphate, after that glucose 6-phosphate, and then glucose. So this is the reverse glycolysis. By the process of the reverse glycolysis, there is a glucose production. So during the one cycle, during the each Kelvin cycle, there is a utilization of 3 ATP and 2 NADPH. So for production of glucose, 1 molecule of glucose, how many cycles it will run? So it requires there is a 6 cycle. So if 6 cycle occur, then it will form 1 molecule of glucose, and 6 cycles means what will happen? Means 6 cycles means 18 ATP and 12 NADPH. Means for production of 1 molecule of glucose, there is a requirement of 18 ATP and 12 NADPH during the C3 cycles. So this is the example of this is the Kelvin cycles, and in the production of Kelvin cycles 1 molecule of glucose is forms. So that's it for Kelvin cycles.